Welcome back or welcome to BioBonanza. In this video, we're going to be talking about biological molecules. Let's get started. But first, what is a biological molecule? A simple definition is that it contains bonds between hydrogen and carbon. This means that the base of the molecule is a hydrocarbon. Added to this hydrocarbon are certain groups of atoms that alter the chemical personality of the molecule, which is how it acts. The first molecules we're talking about are carbohydrates. Carbohydrates can be monosaccharides, disaccharides, and polysaccharides. Monosaccharides. Monosaccharides are a single sugar and they act as fast fuels. A few examples are glucose, fructose, and galactose. Disaccharides. Disaccharides are two sugars bonded to each other. The bond between them is called a glycosidic link. Just like monosaccharides, they are fast fuel, and a few examples are lactose, sucrose, and maltose. Polysaccharides. Polysaccharides are many sugars bonded to each other. They can either help by storing energy or adding structure to something. Two storage polysaccharides are starch and glycogen. Glycogen is found in animals and stores energy for a short period of time. Starch is found in plants and it's able to store energy for a long period of time. These polysaccharides have weaker bonds between the sugars, so they can easily be broken for their energy. Two structural polysaccharides are cellulose and chitin. Cellulose is found in the cell walls of plants, and chitin is found in the cell walls of fungi and also the exoskeletons of insects. Unlike the bonds between sugars in starch and glycogen, these bonds are harder to break. Lipids. Lipids are made up of 3 fatty acid chains and a glycerol molecule. The way these chains are bonded determines whether the lipid is unsaturated or saturated. If there are no kinks caused by double bonds in the molecule, the lipid is saturated, which means it has the greatest possible amount of hydrogen in the molecule. If there are kinks from double bonds, then the molecule is unsaturated since it doesn't have the maximum amount of hydrogen. Saturated fats are easily packed together, so they are solid at room temperature, like butter, and unsaturated fats are not easily packed together because of the kinks in the fatty acid chains, so they are liquid at room temperature, like oil. Lipids have many jobs like protecting things such as organs, insulating things, and aiding in brain and nerve function. They can also store energy and waterproof things since water isn't attracted to them. Other types of lipids are phospholipids and steroids. Phospholipids are made up of only 2 chains and also contain a phosphate group. This phosphate group causes the head of the molecule to be attracted to water, unlike the chains that are not attracted to water. This causes phospholipids to arrange themselves in a way where the heads are facing water and the tails are not. This is how things like cell membranes are formed. Steroids are 4 hydrocarbon rings bonded together, and they act as messenger molecules. One example of a steroid is cholesterol. Proteins. Proteins can have many functions, but this function is determined by how the protein is shaped. All proteins are built up from amino acids, and in an amino acid is an amino group and a carboxyl group. There are 20 different amino acids that cells utilize, and they all contain these groups. Now the structure. The 4 structural levels for proteins are primary, secondary, tertiary, and quaternary. Primary structure is the sequence of amino acids in the chain and the length of the chain of amino acids. Secondary structure is caused by the attraction between hydrogen and the amino acids. This causes the polypeptide, which is the chain, to fold into either an alpha helix or a beta pleated sheet. On the left is the beta pleated sheet and on the right is the alpha helix. Tertiary structure is the random and non-patterned folding of the polypeptide. This folding is caused by the R groups in the amino acid, which is the unique part of the amino acid excluding the amino group and the carboxyl group. If the protein is able to do its job alone, then its structure stops here. Quaternary structure is where proteins are combined together by slightly changing their structure. This is only done if the proteins work together to perform their job. Protein functions include acting as hormones and signaling things, storing things, and finally, acting as receptors. Proteins can also transport things, act as contractile proteins, defend things, and act as enzymes. Nucleic acids. Nucleic acids include DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. These molecules are made up of three things: a phosphate group, a sugar, and a base pair. The sugar for DNA and RNA differ, as deoxyribose, which is found in DNA, has one less oxygen, and ribose, which is found in RNA, has one more oxygen. The base pairs include cytosine, guanine, adenine, uracil, and thymine. Thymine is only found in DNA, and uracil is only found in RNA. Cytosine bonds to guanine, and adenine bonds to uracil or thymine. The role of nucleic acids is to carry and transmit genetic information. This information signals for certain things to happen within a cell. Thanks for making it to the end of the video. Like and subscribe if you enjoyed, and comment down below any video topics you might want me to cover in the future.